is Sarah, and I will be presenting on pesticides, especially neonicotinoids. Pesticides decrease olfactory, learning, and memory in Apis mellifera after exposure to neonicotinoid imidacloprid. So first I'll be introducing you to the topic, then I'll be giving background information, then the impairment of olfactory learning, the effects of imidacloprid on olfactory learning, my conclusion, and the importance and significance of this project. Honeybees have been in a state of decline due to the use of chemicals in agricultural settings. Imidacloprid is a neonicotinoid that is used in many agricultural practices. Imidacloprid has a severe effect on olfactory learning and memory for honeybees. It is important to know what negative effects imidacloprid cause to better understand ways to reduce usage or even remove neonicotinoids from agricultural practices. So what is Apis mellifera and why do we study them? Apis mellifera, also known as the honeybee, are model organisms. They are a key model organism due to their complex social structure, communication methods, behavioral plasticity, and more importantly, their agricultural presence. What is olfactory learning in honeybees and why is it important? Olfactory learning in honeybees is their ability to associate specific smells or odors with reward like pollen and nectar. Olfactory learning is important in foraging behaviors because it allows bees to learn and remember what flowers have the best nectar sources to help sustain the colonies. What are neonicotinoids? Neonicotinoids are insecticides used to kill insects in an agricultural setting. There are also pesticides that are absorbed into plants and can be mainly found in the pollen and nectar of flowers. What is neonicotinoid imidacloprid? Neonicotinoid imidacloprid is an insecticide widely used that disrupts the insect's central nervous system, which in turn affects olfactory learning in bees. So my research question is, how does exposure to neonicotinoid imidacloprid decrease olfactory learning and memory in Apis mellifera? Research has shown that imidacloprid decreases olfactory learning. Additionally, it is one of the most toxic neonicotinoids out there. Imidacloprid specifically affects and disrupts antennal lobes, which are important for olfactory learning. So the effects of imidacloprid on memory formation via olfactory learning. Memory formation is very important because it helps honeybees associate specific odors with certain flowers and pollen/nectar sources. Imidacloprid exposure has effects on short-term memory and long-term memory formation. This figure, especially A and D, showcase the effects of imidacloprid on memory. This experiment that is showcased here uses mass conditioning and space conditioning. Mass conditioning is basically continuous exposure to imidacloprid, and space conditioning is intervals of exposure. I want to focus on A and D, which are the effects of imidacloprid. Imidacloprid had an effect on both short-term memory and long-term memory in both types of conditioning. The striped bars represent memory after 10 minutes, so short-term memory, and the gray bars represent memory after 24 hours, so long-term memory. In both mass and space conditioning, we can see a decrease in both short-term memory and long-term memory. However, we can see a much less in space conditioning. We can see the decrease in the second and third trial, and then we see it a lot in the third trial on D after 24 hours of exposure. So effects of imidacloprid in odor discrimination via olfactory learning. Odor discrimination helps bees discriminate between flowers to determine which are common pollen sources they usually forage from. Imidacloprid has an effect on olfaction and can disrupt associative learning of flower scents, which causes bees to not be able to distinguish between flower sources they have already previously visited. This figure, also from the mast and space conditioning trials, showcases the effects of imidacloprid on odor discrimination, specifically on olfactory recalls. So focusing on A and D as well, because these represent imidacloprid. Imidacloprid had an effect on olfactory recall in both types of conditioning. The stripe bars represent memory of the recall of the flowers after 10 minutes, and then the gray bars represent after 24 hours. And you can see that there is a sharp increase in the mast conditioning, which is A, and we can see a little bit of decrease in the D, but it's mostly the same for space conditioning. Just as a side note, the B and C are two different B and A and B are different neonicotinoids. So A is going to be imidacloprid, B is going to be a different one, and then C represents the usage of both. Effects of imidacloprid on learning and learning performance of olfactory learning. Performance learning in honeybees showcase performance of bees to be able to perform tasks such as odor discrimination. Honeybees who were exposed to imidacloprid were found to have impaired olfactory learning performance. This figure showcases the effects of imidacloprid performance. This shows a control group, the empty circles, and imidacloprid treated bees, closed circles. This figure uses proboscis extension reflex, which is used to measure performance of bees to react to pollen sources. We can see that when exposed to imidacloprid, performance learning decreases by the second and third trials, and we can see that in the black here. So in conclusion, neonicotinoid imidacloprid has been found to have negative effects on olfactory learning. Through research, it was found that exposure to imidacloprid decreases olfactory learning, especially affecting short-term and long-term memory, odor discrimination, and performance learning. So why this is important? It is important to understand how important olfactory learning is in bees. It influences foraging behavior through odor discrimination. Remember when flowers were good pollen sources. It is equally important to know how imidacloprid negatively impacts olfactory learning. And it is important to understand that if we continually expose honeybees to this pesticide, we could lose a vital part of our ecosystem and important presence in agricultural practices. So my references. Thank you.